Transcription is a process that's going to copy just certain sections of the DNA into specific RNA complementary copies. And so if you'll recall, we do have a lot of DNA that contains lots of different genes inside of our cell. So I'm just kind of summarizing one chromosome here with this black line. And from that, we're going to have really hundreds of genes on a single chromosome. And there will be just certain sections of it that would get copied into RNA molecules. So I'm kind of writing those here in red just to indicate that we are just going to be copying sections of the DNA into RNA. And those sections that we're going to copy would be the sections that really we want to make a product from. And when we talk about making a product from it, generally speaking, we're talking about protein, making some type of protein that's going to function inside the cell. Sometimes we do just want to make an RNA molecule from it, but for the most part, it is going to be the protein that we're considering. So one important thing that goes along with the transcription process is really how do we choose or how do we select those sections that we're actually going to copy. So we want to take a closer look at really a bigger region of the DNA. And this would include the portion that we want to copy into RNA, but it's also going to include some other areas in the DNA that will help us to select those specific genes. So here we have a closer look at the DNA, and we're looking at some of the different sections of the DNA that we would have involved in the transcription process. So first off, I want to start off by emphasizing this area which is shown in red here. And this is showing us where we're going to actually start copying into RNA. So this would, what's shown in red, would actually get copied into the RNA molecule. And so we start actually transcribing at what we call the +1 site. So this is where transcription actually begins. And everything gets numbered then according to that. So anything further down, so if something was to this side, we would call it downstream of that particular start site. And then if anything is in this direction, we would call it upstream. And so downstream would get positive numbers and upstream is going to get negative numbers. So if we focus more now on what's shown in green here, what is being shown in green is what we refer to as the promoter region. And the promoter region, its job is to help with that whole selection process. So helping the cell to decide which genes we actually need to make a copy of RNA from. And so there are specific sequences, so orders to the nucleotide bases that we find in that particular promoter region that will help the cell to make that type of decision. There are certainly unique parts to promoters for certain genes, and then there's other things that are pretty consistent within promoter sequences. So the two that I'm showing kind of highlighted in yellow here, those would be sequences that we do find in most promoter sequences. And here we are looking specifically at prokaryotic organisms. So we will have some different sequences once we start talking about eukaryotic promoters. So this is going to be the prokaryotic one. So for the first of what we call the consensus sequences, so each of these, the -35 and the -10, those are what we call consensus sequences. For the first one, the -10, that is what we tend to think of as the, we call it the TATAAT sequence. So that is specifically the sequence that you find in that particular location. The overall job of that particular consensus sequence, if we just write it down below, this one's overall job is going to be to help open the helix up. So we are going to have to break some hydrogen bonds so that we can actually make RNA off of the DNA template. So the -10 consensus sequence, the TATAAT, is going to help us split the strands apart from each other so that we do end up with what we call a transcription bubble. Okay, so we'll open the helix up. That will help with it to make that transcription bubble. The other sequence, the -35 sequence, that one has a different order to the nucleotide bases. In this case, it's going to be TTGACA. And this particular sequence is really, if we write it down below, that one is where the big enzyme complex is actually going to bind to the DNA. And for prokaryotic cells, this enzyme complex that we're talking about is referred to as the holoenzyme. Okay, so obviously two important consensus sequences. One of them is going to help get or recruit the holoenzyme to this particular gene, and the other one is going to help us to open up the helix so that we will be able to actually begin the transcription process. When we talk about the process of transcription in prokaryotic cells, the enzyme that's going to be actually making the RNA is what we call prokaryotic RNA polymerase. So this is the only polymerase that the prokaryotic cells actually utilize, but the enzyme itself does have a total of 5 subunits that make it up. So that's what we're seeing here in this diagram, is that you do have 5 subunits that make up the whole enzyme. So when we do have all of the 5 subunits together, we do refer to it as holoenzyme. And holoenzyme would be the part that is actually going to assemble on that -35 consensus sequence that we just looked at in the promoter region. Once the enzyme is assembled on the -35 site, this particular one, the sigma, is going to leave. So that one does leave, and when it does leave, we're left with just 4 subunits. We'll have the 2 identical alpha subunits, and then we'll have beta 1 and just plain beta. And those 4 subunits are what we call the core enzyme. And it is the core enzyme that actually performs the transcription process. So that will be the enzyme that actually produces the RNA copy at the very end. It all stays together until it reaches the very end of the region that we want to copy into RNA. And at that point, that core enzyme would disassemble.